So today we are going to look at the cerebellum. Very important. The cerebellum. It is located in the posterior cranial fossa of the skull. You have to get time and look at the skull. The skull has an anterior cranial fossa, a middle cranial fossa, and a posterior cranial fossa. So the cerebellum is contained within the posterior cranial fossa, which is found in the occipital bone. And it is inferior to the occipital lobe of the cerebrum. And it is separated from that occipital lobe by a transverse fold of dura mater known as the tentorium. Cerebellum literally means little brain. And as I said, it is located inferior to the occipital lobe. This is the occipital lobe. So it is located inferior to the occipital lobe. And between the cerebellum and the occipital lobe, we have a transverse fold of dura mater in this region. That transverse fold of dura mater between the occipital lobe and the cerebellum, located in this region, is known as the tentorium cerebelli. The tentorium cerebelli. And anterior to it, you have the brain stem, which will be our next lecture after cerebellum. So superior to it, you have the occipital lobe and the tentorium cerebelli. Anterior to it, you have the brain stem. The cerebellum is connected to the brain stem by 3 bundles of axons, and we shall see them. This cerebellum is connected to the brain stem by 3 bundles of neurons, and those bundles are the ones which are known as cerebropeduncles, as we shall see. So you can see our cerebellum, and anterior to it we see our brain stem, which is made up of the midbrain, the pons, and the medulla. This is the brain stem, and posterior to the brain stem is the cerebellum. So these bundles, we have what we call the middle cerebellar peduncle, the inferior cerebellar peduncle, and the superior cerebellar peduncle. These ones, superior cerebellar peduncle, middle cerebellar peduncle, and inferior cerebellar peduncle, are the 3 bundles of neurons which connect the cerebellum to the brain stem. The superior cerebellar peduncle connects the cerebellum to the midbrain. These cerebral peduncles are found within the midbrain. So whenever you see cerebral peduncles, we are in the midbrain. So the superior cerebellar peduncle connects the cerebellum to the midbrain. The middle cerebellar peduncle connects the cerebellum to the pons, and the inferior cerebellar peduncle connects the cerebellum to the medulla oblongata. So that is how our cerebellum is connected to the brain stem, which lies anterior to it. The general functions of the cerebellum, number 1 is to coordinate movement. By the word coordination is general. But here we are saying that the cerebellum controls the timing and the pattern of muscle activation during movement. The meaning of that is that you can say that I am going to perform this action with this frequency. So it determines the time within which you are to perform an action. Or you can say, I'm going to perform this action in the shortest time possible. So it is the one which controls the time of the movement. And during that particular movement, it is the one which determines which muscle is to be activated after another, so that the movement is smooth. Which muscle is to be activated after another. It is very important in the maintenance of equilibrium. There is a way it coordinates the strength of your muscles so that you balance. It does this by connecting to the vestibular system, which contains the inner ear, the vestibular nucleus of the medulla oblongata, and other structures related to balance. So you find that when you are balancing, your muscles have to contract. Some muscles have to relax, others contract, so that you balance. So the organization of those muscles in order to balance your body is mediated by the cerebellum. That is why you lose that sense of balance once the cerebellum has been affected. It controls the tension within the muscles. It determines the load your muscles are going to overcome, and controls muscles such that the tension within that muscle is directly proportional to the load which is going to be handled. The strength you use to lift a 50-liter jerrycan is not the same strength you use to lift a 5-liter jerrycan, because the cerebellum looks at the load and only allows the muscle to contain the tension which is needed, which is just enough to overcome the load according to its size. So it has those functions, the major functions, controlling the timing and pattern of muscle activation during a movement, maintaining of equilibrium, and controlling the tone within the muscles. When you look at the cerebellum from the superior view, here we are looking at the brain from the posterior aspect. We are seeing the occipital lobes, and inferior to them you have the cerebellum. So you're visualizing the cerebellum from the superior view here. So in the superior view, the cerebellum is made up of two hemispheres. The left hemisphere, the right hemisphere, separated by a midline structure known as a vermis. You need to follow and pay maximum attention, because when you're doing obstetric ultrasound, they will require you to scan the fetal head, and one of the scans is going to show you the posterior cranial fossa with the two cerebellar hemispheres and the midline vermis. Vermis means worm-like. So this midline vermis is cerebellar tissue, which is in between the left and right hemispheres of the cerebellum. So it is connected by a midline vermis. The two hemispheres of the cerebellum are connected by a midline worm-like cerebellar tissue known as a vermis, as they lie in the posterior cranial fossa. The cerebellum can be divided into anatomical lobes and functional lobes. The cerebellum can be divided into anatomical lobes and functional lobes. The anatomical lobes are the anterior lobe, the posterior lobe, sometimes the posterior lobe is known as the middle lobe, and the flocculonodular lobe. These are the three anatomical lobes of the cerebellum: the anterior lobe, the posterior or middle lobe, and the flocculonodular lobe. Those are the three anatomical lobes of the cerebellum. So we have our cerebellum here. This is the superior view of the cerebellum, and this is the inferior view of the cerebellum. On the superior view of the cerebellum, we will see this vermis, the left hemisphere, and the right hemisphere. The vermis, the left hemisphere, and the right hemisphere. Then we are going to look, I will see a fissure at this point. This fissure is known as the primary fissure. It's known as the primary fissure. This primary fissure is the boundary between the anterior lobe and the posterior or middle lobe. As you can see, part of the vermis is in the anterior lobe and part of it is in the posterior or middle lobe. And the two lobes are separated by a primary fissure, which is visible on the superior surface of the cerebellum. Now, when you turn the cerebellum upside down, then you're going to see the other lobe known as the flocculonodular lobe. This flocculonodular lobe, part of it is contained within the vermis known as the nodule. And these lateral parts look like leaves. They are known as flocculi. That is why this lobe is known as the flocculonodular lobe. So the three anatomical lobes are anterior lobe, posterior lobe or middle lobe, separated by the primary fissure visualized on the superior surface of the cerebellum. Then on the inferior aspect of the cerebellum, we can see the flocculonodular lobe, made up of these two flocculi. Flocculi means leaf-like. And then the middle nodule, which is part of the vermis. This posterior lobe or middle lobe is separated from the flocculonodular lobe on the inferior surface by what we call the posterolateral fissure. By the posterolateral fissure. So the posterolateral fissure is the one which separates the flocculonodular lobe from the posterior or middle lobe, whereas the primary fissure is the one which separates the anterior lobe from the posterior lobe. So as you can see, part of the vermis is in the anterior lobe, part of it is in the posterior lobe, and part of it is in the flocculonodular lobe. Those are the anatomical lobes of the cerebellum. Those are the anatomical lobes of the cerebellum. This structure, the vermis, as I said, it is distributed within the anterior, the posterior or middle, and the flocculonodular lobe. It has different parts. All these are parts of the vermis, that midline structure, which are named according to their structure. Like lingulates, that part is tongue-like. The pyramid, that part is pyramidal. The uvula, that part is tapering. The nodule is kind of spherical all round, and this is part of the vermis, which is contained within the flocculonodular lobe. So we are seeing the vermis here and its different parts: the central lobule, the culmen, the declive, the folium, and when you go on the inferior aspect, you can see the lingula, you can see the nodule, which is part of the flocculonodular lobe. Then we have our uvula here because it is narrow, tapering. Then we have our pyramid. When you make a sagittal section through this, it looks like a pyramid. Then the tuber. All these are parts of the vermis, named according to their shape. And they are distributed throughout the different anatomical lobes. So as you can see, the flocculonodular lobe only contains the nodule. The anterior lobe contains the culmen, the central lobule, and the lingula. And the posterior or middle lobe contains the declive, the folium, then the tuber, the pyramid, the uvula. All these fall under the posterior or middle lobe. So the parts of the vermis are distributed throughout the different anatomical lobes. I've talked about the primary fissure, which is the boundary between the anterior and the posterior or middle lobe. The posterolateral fissure, which is the boundary between the posterior and the flocculonodular lobe. The prepyramidal fissure is between these two parts of the vermis, which are the tuber and the pyramid. When you go back here, you're going to see the tuber is here and the pyramid is there, and then this is the pre-pyramidal fissure. But the major ones are the primary and then the posterolateral fissures. So we have looked at the anatomical lobes, the anterior, the posterior, and the flocculonodular lobes, and where they are located. Then we go and divide the cerebellum into functional lobes. These lobes are demarcated according to their functions. We have the Spinocerebellum. This is the Spinocerebellum, connected to the spinal cord and the muscles of the body. Spinocerebellum. Vestibulocerebellum, in embryological terms, this is envisaged to have been the first part of the cerebellum to evolve. That's why they call it the oldest, for those who believe in evolution. So the vestibulocerebellum, this is part of the cerebellum which carries out the other function of maintaining equilibrium. It's known as the vestibulocerebellum. Coincidentally, this vestibulocerebellum is contained within the flocculonodular lobe. This vestibulocerebellum is contained within the flocculonodular lobe, and it's that part of the cerebellum which takes part in the balance or equilibrium mechanism. Then we have the cerebral or cortical cerebellum. This is called the cerebral or cortical cerebellum because it is connected to the cerebral cortex. And in evolutionary terms, it was the last to evolve. So the functional divisions of the cerebellum, we have the spinocerebellum connected to the spinal cord and the body muscles. The vestibulocerebellum contained within the flocculonodular lobe and is responsible for maintaining equilibrium, and the cerebrocortical cerebellum, which is that part of the cerebellum which connects to the cerebrum through the thalamus. So when we are looking at these functional lobes and where they are located, still you have our vermis and the two hemispheres on each side, cerebral hemispheres. So where is the spinocerebellum? The spinocerebellum contains the vermis except the nodule of the flocculonodular lobe. It contains the vermis and the paravermal areas. When we talk about a paravermal area, those are the areas in the hemispheres which are on the lateral aspects of the vermis, immediately lateral to the vermis. These are what we call the paravermal areas. So the vermis minus the nodule and the paravermal areas constitute what we call the spinocerebellum. So this region is the spinocerebellum. When we go back to our illustration, the spinocerebellum is in the middle here. It contains the vermis and the paravermal areas. That is what we call the spinocerebellum. It connects to the descending tracts, which are going in turn to connect to the muscles. So this is the part of the cerebellum which connects to the muscles, because the functions of the cerebellum involve muscle activity, muscle tone, equilibrium, activation, timing. So this is part of the cerebellum which connects to our body muscles for their control. The rest of the hemispheres, these are what we call the cortical or cerebrocerebellum. This is the cortical or cerebrocerebellum. This cortical cerebellum or cerebrocerebellum are those parts of the cerebellum which connect to the motor areas of the cerebrum through the thalamus, the cortical or cerebrocerebellum. They connect with the motor areas of the cerebral cortex through the thalamus to inform the motor cortex of the adjustments that it should make before executing a movement. So this is what we call the cortical or cerebrocerebellum. And then the vestibulocerebellum is the one which is contained within the flocculonodular lobe. So the flocculonodular lobe contains the vestibulocerebellum. And this vestibulocerebellum is the part of the cerebellum which participates in the maintenance of balance. So spinocerebellum connecting to the muscles of the body, cerebrocerebellum connecting to the motor areas of the cerebral cortex to the thalamus, and vestibulocerebellum responsible for coordinating balance and is contained within the flocculonodular lobe. Those are the three functional lobes: spinocerebellum, cerebrocerebellum, and vestibulocerebellum. Now, let us go to the histology of the cerebellum, the tissue arrangement of cells within the cerebellum. The cerebellum, similar to the cerebral cortex, the cerebellum, its internal structure is similar to that of the cerebrum. It is made up of an outer layer known as the cerebellar cortex. It is made up of an outer layer known as the cerebellar cortex, and an inner white region known as the cerebellar medulla. an outer layer known as the cerebellar cortex, and an inner white region known as the cerebellar medulla. I told you why we have white matter. I told you why we have white matter. In the nervous system. So generally, the cerebellum contains an outer layer, which is the cerebellar cortex, and an inner white region, which is the cerebellar medulla. That is when you make a section through it. Within the cerebellar medulla, we have cerebellar nuclei which are distributed. These areas are the cerebellar nuclei. They are bilateral, both on the left and on the right. So on the inside of the cerebellum, the cerebellar medulla, we have cerebellar nuclei distributed within the core of the white matter of the cerebellum, which is the cerebellar medulla. And when you name this nuclei from lateral to medial, this big one is the one we call the dentate nucleus. This big one is one we call the dentate nucleus. Then we go to this one known as the emboliform nucleus. Emboliform nucleus. Then we have this one known as the lobes nucleus. And then we have this one known as the fastigial nucleus. The dentate, the emboliform, the globose, and the fastigial nucleus. These are the regions of gray matter within the cerebellar medulla, and they are known as cerebellar nuclei. We shall see how they interconnect with other parts of the brain and the pathways within which they are involved. So outer cortex, inner medulla, which is made up of white matter, distributed within the inner medulla are the cerebellar nuclei. Now let us expound on this cortex. It may look a thin layer. This cortex looks as though it is a thin layer, but when you magnify this cerebellar cortex, it is made up of principally 3 layers. The cerebellar cortex is made up of 3 layers. The outermost layer is known as the outer molecular or plexiform layer. Deep to it, we have the Purkinje cell layer or the intermediate layer, because it is made up of these neurons known as the Purkinje cells. The deepest layer of the cerebellar cortex is the inner granular layer. Granular because it has very many cell bodies. When you stay in it, they look like granules. So the inner granular layer, this is the layer which is adjacent to the white matter of the cerebellar medulla. So the cerebellar cortex is made up of 3 layers. The outer molecular layer or plexiform layer. They call it molecular because it has very many synapses and there's a lot of exchange of neurotransmitters there. And neurotransmitters are molecules. That's why they call it an outer molecular layer, because of the extreme number of synapses. Then we have the intermediate or Purkinje cell layer, because it has these neurons known as the Purkinje cells. And then the deepest layer, which is nearer to the white matter, is the inner granular layer. They call it granular because it has very many cell bodies, and when they are stained, they look like granules. So you have the outer molecular layer, the middle or intermediate Purkinje cell layer, and this is the deepest, which is the granular layer. After the granular layer, then you go to the white matter of the cerebellum, which is the cerebellar medulla. Those are the layers of the cerebellar cortex. Those are the layers of the cerebellar cortex. It's made up of 3 layers. Let me see whether I have other illustrations which bring it out clearly. You can see this is the cerebellar cortex, the outer layer, and this is the cerebellar medulla, which is the white matter in the core. And inside the cerebellar medulla, we have the nuclei of the cerebellum. And I've said that this outer layer, which is the cerebellar cortex, can be further divided into 3 layers. You can see this is the cerebellar cortex only. It has the outer molecular layer, the middle Purkinje cell layer, and the inner granular layer. Then you go to this inner granular layer is the one now which is adjacent to the white matter. Now let us look at the details of this cerebellar cortex. We are now in the cerebellar cortex, that outer layer of the cerebellum. Let us look at its detail. You're seeing black arrows. Some are entering into the cerebellum and some are leaving the cerebellum. So this means that the cerebellum receives electrical impulses. That is why you see these arrows entering like that. The cerebellum receives electrical impulses. And then it also transmits out electrical impulses. I repeat, the cerebellar cortex receives electrical impulses, and it also transmits out electrical impulses which are carrying information about its intended plan in regards to motor activity. So that is how the cerebellum behaves. It receives, these ones coming in, we call them afferent. It receives afferent information and it takes out efferent information. So the cerebellar cortex is receiving, that is the afferent information, and also it is transmitting out, that is efferent information. Afferent information, that is information entering into the cerebellum, is transmitted through 2 types of neurons. Afferent information is transmitted through 2 types of neurons. These are known as climbing fibers and Morse fibers. Climbing fibers and Morse fibers. Those are the 2 types of neurons which transmit afferent information towards the cerebellar cortex. Climbing fibers and Morse fibers. They are the 2 neurons through which afferent information reaches the cerebellar cortex. Climbing fibers are known as climbing fibers because they are going to move up to the molecular layer. They are going to move up to the molecular layer and synapse with the dendrites of Purkinje cells. Whereas Morse fibers are going to end within the granular layer. And then they are going to synapse with different neurons of the cerebellum like what we call the granule neurons, the Golgi neurons, as you can see, the granule neurons and the Golgi neurons. Their terminal end has this structure which can synapse with more than one neuron and is known as the glomerulus. So for them, they end their terminal, which is the glomerulus, ends in the granular layer where it synapses with the different neurons known as granule neurons and Golgi neurons. Then these neurons are the ones which are going to transmit those impulses, as you can see, to the molecular layer. So whereas the cerebellum receives afferent information through climbing and Morse fibers, the climbing fibers will move up to the molecular layer and synapse with dendrites of Purkinje cells, whereas the Morse fibers will end in the granular layer and synapse with many granule neurons and Golgi neurons, which will in turn transmit the impulses to the molecular layer. That is how afferent information reaches the cerebellar cortex. How does efferent information exit? Because this one is afferent through the climbing and Morse. What about the one leaving? The one leaving is efferent information. Efferent information is moving out of the cerebellar cortex through axons of Purkinje cells. So this information is moving out through axons of Purkinje cells. So efferent information exits the cerebellar cortex through axons of Purkinje cells. And this information is going to go to the white matter on towards the cerebellar nuclei. So this information is going towards the cerebellar nuclei. And it is inhibitory. That is why it is having a minus. So whereas the information entering into the cerebellum, which is afferent, is excitatory. That's why I have a plus. The efferent is inhibitory, and it is going to inhibit the cerebellar nuclei, which are within the white matter. And this efferent information, on top of being inhibitory, it is transmitted through axons of Purkinje cells. So it's like information coming in, afferent information, will end up exciting the dendrites of the Purkinje cells. You can see those plus. Then when the Purkinje cells are excited, they respond by transmitting out inhibitory information to the cerebellar nuclei. And we saw that neurotransmitter when we are looking at the basal ganglia, glutamate, which is excitatory, and GABA, which is inhibitory. So that is how information runs through the cerebellar cortex. Afferent, which is excitatory to Purkinje cells. Efferent, which is inhibitory to cerebellar nuclei. Afferent through climbing fibers and Morse fibers. Efferent through axons of Purkinje cells. Morse fibers end within the granular layer, whereas climbing fibers reach the molecular layer and synapse directly with the dendrites of the Purkinje cells. Morse fibers synapse with granule and Golgi cells, which in turn will transmit information to the molecular layer, divide into what we call parallel fibers, and these parallel fibers are the ones which are going to synapse with the dendrites of the Purkinje cells. Now, these Purkinje cells, it is not a must that they must always transmit out efferent. Sometimes there is no need of transmitting out efferent information. That is why we need neurons like the basket cells, which are going to inhibit them. So sometimes these Purkinje cells do not transmit out impulses. because of being inhibited by the basket cells. So the basket cells inhibit the Purkinje cells so that they don't transmit out this inhibitory information to cerebellar nuclei, but sometimes they can withdraw the inhibition and they disinhibit them so that they transmit out. So these basket cells are a control to the Purkinje cells, such that they are not always transmitting out inhibitory impulses to the cerebellar nuclei. So they inhibit them and disinhibit them. So they are a control to the transmission of these impulses. Stellate cells, they are star-like, also participate in the inhibition of these parallel fibers. You can see these parallel fibers which are going to excite Purkinje cells, dendrites. So these spiral fibers can also be inhibited by the stellate cells, and sometimes the stellate cells can remove the inhibition so that the impulses are transmitted. So there must always be a control, inhibition of stimuli and disinhibition of stimuli. So whenever they ask you, how does afferent information reach the cerebellar cortex? Through climbing fibers and Morse fibers. What are the differences between climbing fibers and Morse fibers? Climbing fibers will move up to the molecular layer and synapse with the dendrites of a Purkinje cell, whereas Morse fibers end in the granular layer and synapse with many Golgi and granule cells. The granule cells will now ascend in the molecular layer and divide into parallel fibers, which will synapse with more than one Purkinje cell. How does efferent information leave the cerebellar cortex? Through the axons of Purkinje cells. And where does this efferent information go? It goes to the cerebellar nuclei within the white matter. And what type is it? It is inhibitory. Which neurons control the transmission of impulses within the cerebellar cortex? We have the basket cells which can inhibit the Purkinje cells so that they don't transmit out, and then the stellate cells which can inhibit these impulses through the parallel fibers so that they may also not be transmitted. Such that sometimes the cerebellum is giving out efferent information, sometimes it is not giving out, depending on the activity it wants to perform. I've given you very many notes explaining to you the interconnections within the cerebellar cortex that I've just talked about. Afferent, climbing, and Morse. Efferent, axons of Purkinje cells. Now, where do the climbing fibers come from? Because we have said that these climbing fibers enter the cerebellar cortex and move out to the molecular layer. But where are they coming from? When we look at this illustration, this is our brain stem, this is our cerebellum. And we say that the cerebellum is connected to the brain stem through 3 peduncles, the superior, the middle, and the inferior. So we have seen our cerebellum having the external cortex, which has 3 layers that we have talked about, and the internal medulla, which is white matter containing cerebellar nuclei. So in the medulla oblongata, there is what we call the inferior olivary nucleus. The inferior olivary nucleus. So many or most of the climbing fibers originate from the inferior olivary nucleus of the medulla oblongata, and they will ascend as the olivocerebellar tract towards the cerebellar cortex. So these climbing fibers we saw entering, transmitting impulses to the molecular layer of the cerebellar cortex, most of them are coming from the inferior olivary nucleus of the medulla oblongata. They move as the olivocerebellar fibers until they go and reach the molecular layer of the cerebellar cortex. As they are moving towards the molecular layer of the cerebellar cortex, they transmit some of their impulses to the cerebellar nuclei, and then they continue up until they reach the molecular layer of the cerebellar cortex. So they are originating, their cell bodies are within the inferior olivary nucleus of the medulla oblongata. Then these Purkinje fibers, they are the ones which transmit out information from the cerebellar cortex. They transmit that information to the cerebellar nuclei. This information can be inhibitory to prevent the cerebellar nuclei from transmitting out information. And if it is not coming, these cerebellar nuclei are disinhibited so that they continue transmitting out information to the different parts of the brain stem. So these Purkinje cells are either bringing inhibitory impulses to these cerebellar nuclei, or when they are not transmitting because of their control, these cerebellar nuclei are not inhibited, they continue transmitting out information. That is how the Purkinje cells control the deep cerebellar nuclei. So we can see the climbing fibers moving towards the cortex, and then the Purkinje fibers taking out information. They can inhibit, and if they don't inhibit, this nucleus is not going to be inhibited, it's going to continue transmitting out impulses. These impulses going out from the cerebellar nuclei are transmitting information from the cerebellum related to control of muscle activity, like balance, pattern of muscle stimulation, and muscle tone, as we talked about whenever they are required. The inhibitory neurotransmitter is GABA, the excitatory neurotransmitter is glutamate, the same neurotransmitters we saw when we are talking about the basal ganglia. So we can see climbing fiber entering, coming from the inferior olivary nucleus of the medulla. It enters up to the molecular layer of the cerebellar cortex. Then we are seeing here the Purkinje cells transmitting out information towards the cerebellar nuclei. And this information is inhibitory. But sometimes if they are not transmitting out, the cerebellar nucleus will continue being excited because it's no longer receiving inhibitory. You can see the Mose fibers. We are going to see where they are coming from also. The Mose fibers, they also bring in information, but they end in the granular layer. And then the granule cells will pick those impulses and take them to the molecular layer. They also give some of their stimulus to the deep cerebellar nuclei, like the climbing fibers, before they transmit it to the cerebellum. Mose fibers. Where do Mose fibers originate from? We have seen climbing fibers originating from the inferior olivary nucleus of the medulla. What about Mose fibers? Where do they originate from? Mose fibers originate from the motor areas of the cerebral cortex, the pons, the medulla, and the spinal cord. They have a diverse origin. They originate from the motor areas of the cerebral cortex, the pons, the medulla oblongata, and the spinal cord, and they transmit information towards the cerebellar cortex, the granular layer. As we saw. So we are seeing here some muscle fibers bringing information from the pons. They are bringing information from the pons. Some are bringing information from the spinal cord and diverse areas. It is the climbing fibers which were originating from the inferior olivary nucleus. But most fibers bring information all the way from the cortex, pons, and then the spinal cord until they reach the granular layer of the cerebellar cortex. Now, the cerebellum must receive information about what is going on in the entire brain so that it can add its contribution. That is why the cerebellum is receiving information from almost all parts of the brain so that it can integrate that information, make sense out of it, and give its contribution, which is going to promote smooth muscle activity. The deep cerebellar nuclei are the dentate, the fastigial, the emboliform, and the globosus. The emboliform and globosus nuclei are also known as interposed. The emboliform and globosus nuclei are also known as interposed nuclei because they are located between the dentate and the fastigial. So you can see the dentate nucleus is this one. The fastigial is that one. Then these two, the emboliform and the globus is split into two. The emboliform and the globus, because they are between the dentate and the fastigial, that is why they are known as the interposed nuclei. And when you name them from lateral to medial, you have the dentate, the emboliform, the globus, and the fastigial. The emboliform and the globus nuclei are between the dentate and the fastigial, and they are known as the interposed nuclei. So we have talked about these nuclei of the cerebellum, that they receive inhibitory impulses from the axons of Purkinje fibers. But sometimes those inhibitory impulses do not come such that those nuclei continue transmitting information out. They only stop transmitting information out when they have received stimulation, inhibitory stimulation from the Purkinje cell axons. The fastigial nucleus is the one which is concerned with balance, and it is the one which is connected to the vestibular nucleus or apparatus, which is responsible for control of equilibrium. So the fastigial nucleus is very important in that function of the cerebellum known as balance. And you see it is the most medial. The globosus nucleus, this nucleus is split into two. It's this one. This one sends information through the superior cerebellar peduncle to the red nucleus of the midbrain. This red nucleus is very important in controlling the action of flexors and extensors. Whenever you're performing your actions, when the flexors are stimulated, the extensors are relaxed. You remember the rubrospinal tract? So this globosus nucleus, because it is connected to the red nucleus of the midbrain through the superior cerebellar peduncle, it's very, very important in the control of flexors and extensors during muscle activity. There must be a balance between extensors and flexors when you are moving, and it is the globus nucleus which participates in that mechanism by connecting to the red nucleus. You go back and read about the functions of the cerebrospinal tract, and that is where the globus nucleus gives its contribution. Information from the emboliform nucleus is the one which is going to go to the thalamus, and from the thalamus, it is going to go to the cerebral cortex. Information from the emboliform nucleus is going to go to the specific motor nuclei of the thalamus. From there, it's going to be transmitted to the cerebral cortex. And this information participates in the control of precision. Precision is whereby when you determine to perform an activity, when you determine to perform a motor activity, after performing it, your muscles stop moving. They don't continue moving. You say, let me hold a pen, and once you hold that pen, that activity stops because it has achieved the objective. That is what we call precision. So information coming from the emboliform nucleus is going to the cortex, but it passes through the specific motor nuclei of the thalamus, and it is very important in controlling precision in the limbs. Precision in the limbs. And that is why it is also connected to the Spinocerebellum, which connects to the muscles of the body. Information coming from the dentate nucleus, which is the largest, is concerned with voluntary movements. All your voluntary movements, in order for them to be perfect, perfection, I mean that the muscles must have the proper tone and they must be activated in the correct sequence. So that information about proper tone, correct sequence of activation of muscles, voluntary movements, comes from the dentate nucleus. The dentate nucleus sends it to the specific motor nuclei of the thalamus, and then it will go to the cortex. This dentate nucleus also connects to the red nucleus, because when you are carrying out voluntary movements, extensors and flexors must coordinate properly. So precision we saw it, but now for this is the control of voluntary movements so that they are very smooth. The tone in the muscles is the correct one. The extensors and flexors are functioning in coordination. That information comes from the dentate nucleus. It goes to the thalamus, from the thalamus to the cortex, or some of it goes to the red nucleus. So concerned with voluntary movements by making sure that they are smooth and organized. You go and read more about the different functions of the cerebellar nuclei. I said that the cerebellum is connected to the brainstem through bundles of neuron axons. The superior cerebellar peduncle connects to the midbrain, and from there to the thalamus and the cerebral cortex. The middle cerebellar peduncle connects to the pons. The inferior cerebellar peduncle connects to the medulla oblongata up to the spinal cord. Information entering or leaving the cerebellum. Information entering or leaving the cerebellum either passes through the superior cerebellar peduncle, the middle cerebellar peduncle, or the inferior cerebellar peduncle. There is no other route towards or out of the cerebellum except those three. So, the inferior cerebellar peduncle, most of the fibers passing through the inferior cerebellar peduncles are afferent. Afferent means information there is going towards the cerebellar cortex. So most of the fibers passing through the inferior cerebellar peduncle are transmitting impulses towards the cerebellar cortex. So most of them are afferent. That's why I say it is predominantly afferent. Most of them are afferent. And that information which is passing through the inferior cerebellar peduncle to the cerebellum, most of it is coming from skin, from deeper structures, and for balance, like the inferior vestibular nucleus. So information is coming from the inferior olivary nucleus and being transmitted through the inferior cerebellar peduncle. And I say that most of that information passing through the inferior cerebellar peduncle is afferent. Some of the efferent information which is passing through the inferior cerebellar peduncle is this information, because I say that some information from the Purkinje cells going towards the vestibular nucleus. So this is some of the efferent information, whereas most of the information is afferent, but some Purkinje cells transmit efferent information through the inferior cerebellar peduncle towards the brain stem. So when we say predominantly, it means that there are some efferents which pass through it, but most of the information entering through the inferior cerebellar peduncle, most of it is afferent. It is going towards the cerebellum. The middle cerebellar peduncle also is predominantly afferent. Most of the information passing through it is entering into the cerebellum. And I said that most of the fibers of the middle cerebellar peduncle are commissural. The middle cerebellar peduncle of the right side is transmitting fibers from the left, and the middle cerebellar peduncle of the left is transmitting fibers from the right. But it is predominantly afferent. Afferent means that most fibers there are being transmitting information into the cerebellum. Now, the superior cerebellar peduncle is the one which is predominantly efferent. Most of the fibers through the superior cerebellar peduncle are transmitting information outwards. So it is predominantly efferent, much as it has some afferent information, but it is predominantly efferent, and it's mainly transmitting impulses to the brainstem, the thalamus, then on their way to the cerebral cortex. So those are the peduncles of the cerebellum. Now I am going to task you to read about the afferent tracts which are moving towards the cerebellum. There is what we call the dorsal spinocerebellar tract. We even talked about it when we were looking at ascending tracts. This is one of the tracts which is transmitting afferent information towards the cerebellum. I'm not going to talk about it again. You go and read about the posterior or dorsal spinocerebellar tract. And I've given you a very good illustration here. You can see the dorsal spinocerebellar tract transmitting information through the inferior cerebellar peduncle to the cerebellum. That one you go and revise it. Then there is the anterior spinocerebellar tract. We also talked about it. That anterior spinocerebellar tract, which is transmitting information from the contralateral side, and then it is being taken to the cerebellum through the superior cerebellar peduncle. Cranial cerebellar tract This one is coming from the nucleus cuneatus to the cerebellum. I also talked about it. It is also an afferent tract moving towards the cerebellum. You'll go and read about that tract also. This is how afferent information reaches the cerebellum. Olivocerebellar, which I talked about, where climbing fibers pass from the inferior olivary nucleus to the cerebellum, the olivocerebellar tract. From the inferior olivary nucleus, this olivocerebellar tract is going to move cross to the opposite side. It is going to pass through the inferior cerebellar peduncle and reach the cerebellar cortex. Pontocerebellar tract from the pons to the cerebellum. And I've given you that tract from the pontine nuclei. It crosses to the opposite side, passes through the middle cerebellar peduncle, and then enters the pons. Tectocerebellar tract from the midbrain, because the midbrain, that's where you have the superior colliculus, which is the tectum, and it passes through the superior cerebellar peduncle and reaches the cerebellum of the same size. This is the tectocerebellar. From the tectum, then it passes through the superior cerebellar peduncle and then enters the cerebellum. These are some of the afferent fibers passing through the superior cerebellar peduncle. Trigeminocerebellar tract from the mesencephalic nucleus of the trigeminal nerve. From the mesencephalic nucleus of the trigeminal nerve, it ascends towards the cerebellum through the superior cerebellar peduncle of the ipsilateral side. So this illustration is showing you all these tracts that I've mentioned, the ascending and then the efferents. The efferents, we have the efferents are the ones coming from the cerebellar nuclei, because information coming out of the cerebellar nuclei, information coming out of the cerebellum originates from the cerebellar nuclei. So the efferent information is that information coming from the deep cerebellar nuclei moving out. This one, fastigiobulbar, is moving from the fastigial nucleus to the reticular formation of the brain stem. And I've indicated it on this side. This is the efferent information, and you see most of it is passing through the superior cerebellar peduncle. Cerebelloreticular tract from the cerebellum, especially from the emboliform nucleus to the reticular formation of the brain stem through the superior cerebellar peduncle. Then the cerebello-olivary from the emboliform nucleus and globus nuclei. That information moves out and goes to the inferior olivary nucleus, and so on and so forth. Dentothalamic from the dentate nucleus to the specific motor nuclei of the thalamus of the same side, and then on towards the cerebral cortex, because once it is going to the thalamus, it's on its way to the cerebral cortex. Globus emboliform rubro from the globus nucleus and emboliform nucleus, rubro for red nucleus, to the red nucleus of the brain, of the midbrain. Fastigiovestibular from the fastigial nucleus to the vestibular nucleus of the brainstem for the control of equilibrium. And I've finally talked about the different functions of the different functional lobes of the cerebellum.